All right, everyone, we're going to get started with the next session. If I don't start on time, I'm in big trouble with Catherine Forsey, and nobody wants that. So I'm going to start on time. So this session is about CMT-SORD, and I'm joined here in this panel by Dr. Stefan Zuckner and Dr. Riccardo Perfetti. And I'm going to give it just a brief overview and talk about setting the stage for the story and the role in particular that the CMT community has played and the CMTA, because it's been a really great story. And we think it's going to be the story potentially of the first product approved for any CMT subtype. We make investments, as I mentioned earlier, in really foundational aspects. If you think of this like building a house, drug development like building a house, we've invested in a strong foundation so that when the science is ready, the foundation is there. So natural history studies, outcome measures, and the outcome measures that you'll hear Dr. Perfetti talk about, those are things that we help develop in collaboration with the Inherited Neuropathy Consortium so that, again, when the drug is ready, we're ready too. We also invest in the Genesis Project, which Dr. Zuckner is heading up, and that's really the framework for drug development. That's what helps it go quickly. And I talked this morning a little bit about the steps in drug development. And what's incredible is this story, as opposed to most drug development where you start with a gene discovery and you get to phase 3 efficacy data, that's that really big study that gets you a drug approval, that can take more than 10 years, 10 to 15 years from beginning to end. And what's incredible here is that there's a company, Applied Therapeutics, who's our sponsor for this year's summit, who had the biology already going. So we talked about genetic stories and biology stories. This is a story about how genetics can go from 2020, finding a gene and publishing that story, to starting to enroll that efficacy trial, the phase 3 trial, 2 years later, to getting data from that efficacy study in 4 years. That is unprecedented. As someone who's developed drugs, that is a phenomenal story. And so Dr. Zuckner and Dr. Perfetti will take you through the science. But from a timeline perspective, Applied was already working on AT-007, also called govorestat, when 2020 came along. And what we find is sometimes there's a patient here, there's a patient there, there's a patient here. And the Genesis Project can bring it all together. It turns out there's a whole community of patients, and it's called SORD. And because the gene was related to some biology that was already ongoing, we were able to fast forward in 2020 and 22 to get those trials enrolled. Patients as partners in research played a critical role in both enrolling that phase 1-2 trial as well as getting that phase 3 efficacy trial enrolled as quickly as possible. We also created some focus groups to make sure that the voice of the patient was part of the drug development process. And we've really worked closely with Applied throughout this process so that in 2024, when they got the first look at that phase 3 data, which looked really exciting, we were able to jump into action one more time. And so when they got that data, there's a pathway called the accelerated approval pathway, where you can take that data and ask the FDA to consider you for approval. And so our own Vittorio Ricci actually started a change.org petition, and we helped market that. You may have seen that in our social medias to say, hey, FDA, there's nothing out there for CMT folks. This is a rare disease. Will you please consider having a conversation with Applied about moving us into accelerated approval? And we got 10 signatures and 100 signatures and 1,000 signatures. And then we said, wouldn't it be cool if we got more than 3,300 signatures, which is the number of patients with SORD in the U.S.? We ended up with 4,000 signatures on that petition. Vittorio sent it to the FDA. Applied has been working with the FDA, and they've agreed to actually consider an application for accelerated approval. So that advocacy really got us to the next step. And so we are excited about the possibility that CMT-SORD means for the community. Getting one across the finish line means that we have a regulatory pathway that can be used in other types of CMT. We're grateful for the community. And I'm now going to turn it over to Stefan to share the beginning of this remarkable scientific story.
Great to be here. It really is. Also, I met several of the SORD patients here last night, this morning, and it just really gives me goosebumps. Sort of that our work, working in the lab, behind a computer, they're not patients there or people that remind you what you do. So it's good to meet you all. So as Sue said, this is an incredible story. So we want to highlight it because it sort of shows the principal pathway we have in mind that Sue talked about this morning. And from a genetics perspective, what really matters is for geneticists to be successful is to compare genes and genomes from one person to another person, from one patient to another patient, and ideally also patients from different countries with different ancestries. All that helps us to really interpret the data. I'm not going to go into this, but the genome is a huge place. So finding this little, this one piece that goes wrong is very hard. So we need a lot of data to make sense out of it. And there's the Genesis database that is also supported by the INC and the CMTA. Without this, it wouldn't be there. We have now nearly 3,000 genomic data sets from CMT patients. Many of those don't have a diagnosis yet. So this is a sort of a super important resource we have, and we want to keep growing. So you're all welcome to participate. When we found SORD, it was very exciting for the lab, of course. Even though we find it probably about 2 new CMT genes per year, we knew pretty quickly this was special. This was different. Here you see the set of the initial families, and I just want to show you also because on top you see these numbers there. That's sort of the postal address of the most common change in SORD. And when you look at these family trees or pedigrees, you probably notice that. So the dark means there is that's the person with CMT, and the non-dark symbols are healthy individuals. That many of the CMT patients, their parents didn't have anything, and also their parents hadn't CMT. Also, their siblings didn't have CMT. So we call it sporadic occurrence. And those are patients that are challenging sometimes for CMT doctors to make a call. Is this CMT, or is this something that comes from another source, like diabetes or an inflammation or something else that has just nothing to do with genes? So we especially pleased really to be able to diagnose these sporadic patients, and many of them. Of course, over time, we found additional changes. Here's sort of a schematic of that. And that work continues. There are people being screened in clinical labs and also in research studies all over the world, really. And that enriches our knowledge about this particular gene. So this is a bit technical. I'm breaking it down super simple for you, I hope. The red box, just focus on the red box for a second. So when we find a novel disease gene and we're starting to try to convince us, is this really a CMT gene? The first question that somebody like Ricardo from pharma industry would ask, well, what is going on there? Do you lose this gene, or is it just, does it change its function? What is going on? And in this case, in this blue box within the red box, you see that basically on the upper, you don't see these dark bars. So something is missing there. So pretty quickly, by studying CMT patient samples, we understood that when you carry this change, you really lose SORD, you lose the gene, and you lose the protein. So no SORD protein is being made. It sounds like for you maybe like, oh, okay, that's pretty logical. But often that's not the case, and it's way more complicated. So that was great. There was a clean answer. When you have this mutation, you don't make the SORD protein. And when you don't have the SORD protein, obviously the protein cannot function in its normal way. And when it doesn't function, then what the SORD protein really does, it takes care of a chemical substance. we call sorbitol, and it removes sorbitol. So sorbitol normally is almost not measurable. When you look at the green box, you see these dots there, and a group of dots that are much higher. These that are much higher, they are from patients. So you can easily see that patients have a lot more sorbitol because they cannot get rid of it because they don't have the SORD. So within really a few months, we figured out what's going on here. You have a mutation in this gene, then you lose the protein, no more protein. Without the protein, you cannot get rid of sorbitol, so your sorbitol is super high. And that all happens in what we call a pathway. So when certain proteins, they work together, they don't work for themselves. And it turned out that this pathway had been studied for 20 plus years to understand diabetes and diabetic neuropathy better. And that where also Applied Therapeutics had a big interest in. And essentially, this is what I just explained you in the schematic again. You don't have sorbitol dehydrogenase. This is the fancy name for SORD that we use. So you get the higher sorbitol. And then the idea is, well, how do we prevent getting the sorbitol high? Because obviously it's linked to the disease. And one idea is, can we block the protein in front that actually creates sorbitol in the first place? And I think Ricardo will talk about that, because that's the idea of the drug, really. So again, technically, I'm not going to go into this, just showing you with some graphs here. What we then often do is we try to confirm these things in an animal. What is that? 1 minute or one beer? So animals, in this case, fruit flies, and forgive me the technical term, Drosophila melanogaster, that's fruit fly, okay? That's literally the fruit fly in your kitchen, okay? And believe it or not, we can make them have CMT. So why we do this? Because it's cheap. We don't have any ethical problems in squishing them, and they have a short life, right? So we can study this fast. This is very important, by the way. When we do mouse studies or rat studies, which we do, they take longer, and then you have to wait longer. This was all about speed. So they only live like 60 days or so. But anyway, these fruit flies develop, these SORD fruit flies, they develop a form of CMT. And we were able to treat them with the drugs that Applied Therapeutics is developing. And sure enough, these flies, we could cure them of CMT. Kind of crazy when you think about it. But all that data helped us to discuss with Applied Therapeutics, convince them, and they were convinced that maybe we should move forward with the trial. And just a couple more slides, sorry, Catherine. We are keeping screening worldwide the patients with CMT to find more SORD patients. You can see. We also, meanwhile, have created, which took a lot longer, an animal model, a bigger animal model, a rat model. And in fact, this is a great model because it also clearly shows CMT. So now we have an animal model, because there are many questions still open. Even when the drug is approved, hopefully, there will be many scientific questions that we still want to answer around this type of CMT. So this will be very helpful. And I, of course, want to acknowledge especially Adriano Rebello, Andrea Cortese. They are both postdocs, and Andrea was a trainee in my lab at the time. Without them, it probably wouldn't have found it, or at least not as quickly. So thank you.
Thank you, Stefan. Thank you, Sue. And it's a great opportunity to be presenting how we took the science developed by the group of Stefan and all of his collaborators to a clinical development program relatively quickly. I think the reason why we were so fast we knew the pathway from a biology point of view, because this pathway is very critical for also other medical conditions. But also, I guess, because we are a small company, very agile, and also very open on listening, learning, and interacting with the academics, but also with the community of patients. What you see in this slide is a little bit a recap on the specificity of CMT-SORD. So this is an autosomal recessive disease that leads to the absence of a critical enzyme that transforms sorbitol into fructose. This pathway has a very marginal activity in healthy individuals. Only about 3% of glucose gets into that. But in patients that are deficient for SORD, the accumulation of sorbitol, this toxic, becomes very, very critical. You'll see that in the next few slides. So this is a pictorial. I know not all of us like biochemistry, but this is a very simple schematic. So glucose is the substrate for energy production. The way we work is primarily due to the fact that we can burn glucose. It's the fuel that allows us to be functional. So most of glucose needs to produce energy. Very little one goes into a stream that for us as humans have no gain. So we don't make anything out of that little stream that is 3% of the overall glucose. There are only two steps on that stream. One is governed by an enzyme called aldose reductase, and the other one is called by the enzyme that is defective in patients with SORD deficiency. Because patients that are defective on SORD deficiency cannot process sorbitol and make fructose, they have an accumulation of sorbitol. So we want to block that excess of sorbitol. Okay. So how do we design a study that translates the potential for the intervention that blocking sorbitol delivers on outcome? And what is the outcome? Okay, well, in metabolism, so this is a metabolic pathway, you're blessed by the fact that you could measure whether you have really modified the excess of that toxic. So you could measure that toxic to see whether you have reduced the level of toxic. But in clinical medicine, you also need to show that that as a value produces a benefit. But then what is the benefit that matters? Well, this is no longer the opinion of physicians and scientists. It has to be also the opinion of patients, right? If I change something, am I certain that that change will be beneficial to you as a patient? So we as a company have roundtables for patients to discuss what exactly is the manifestation of the disease for you. What component of the disease are the most critical one? Where are you hoping for changes? Because that allows you to define a study that has the endpoint, the measures that matter. Because that is critical. You don't want just to work on the biology. You want to produce outcome. So the outcomes that are more important here is the biochemical, are reducing the toxic. Are you allowing patients to function better? So functional outcome. Are you making them well-being overall better? Patient-reported outcome. So what you see here is a big study that we designed with different checkpoints where we were evaluating whether we reduce the level of toxic, but also we made patients more functionally active, and are they overall feeling better? So chemistry, functional outcome, patient-reported outcome. Already, very interestingly, what we learned looking at patients before we gave them the first dose is that the level of this toxic was not the same among all of them. So the level of this toxic was greater in patients that had more severe manifestation, was lower in patients that was evolving slowly, and the manifestation was less severe. That indirectly perhaps suggests that lowering may actually provide benefit. And the first thing we did, again, was check the biochemistry. What you see in this graph in green is the reduction that we saw in patients that were exposed to the drug. And in gray, you see the reduction in patients that were exposed to placebo. So we were able to effectively reduce the level of the toxic in patients that were exposed to the drug, which is called govorestat. And this was overall generally safe in these patients. This was done at month 3 of the study. The study continued to go. And at month 12, we start measuring how patients function. So there are several tests of functional outcome, how fast you can run, how quickly you can stand up. So a composite of this endpoint was measured versus the level of sorbitol. What you see on the graph on the left is the greatest was the reduction, so the vertical assay. The lower was the level of sorbitol. So what this shows is that patient for which we reduced the most had the greatest benefit, indicating the effect of the functional outcome. So the first step was reducing the toxic. The second one was to see whether reducing the toxic makes the patient function better. But perhaps even more important was how do patients feel? So here you have several different metrics on mobility, on fatigue, on lower limb mobility, upper limb mobility. Patient invariably, as you see on the green part of the slide, patients that got the active treatment felt better. And this was at 12 months. So overall, what this study shows is that reducing sorbitol matters. Reducing sorbitol improves the functional outcome of patient, but also has an impact on their overall well-being. The study has obviously been successful so far. This is still an experimental drug. We continue to interacting with organizations like yours, with regulators, and we are very much focused on moving this very, very fast as we did in the past. Thank you for your attention.